Thanks, Tessa. I am Samira. I am a software engineer at a biotech company called Nucleases. My educational background is in molecular biology, and I'm going to talk about gene editing, specifically therapeutic gene editing. So the promise of gene therapy, which was this idea that people got very excited about starting in the late 80s and early 90s, was that we could cure diseases by modifying a patient's DNA. And there's a lot of applications for this. There are over 3,000 well-studied monogenic disorders, meaning they're caused exactly by one gene. My microphone approaches me. There are over 3,000 well-studied monogenic disorders caused by exactly one gene. Things like muscular dystrophy, cystic fibrosis, cancer-causing disorders. And theoretically, in principle, we know how to fix these. And what I mean by that is that we know for each of these diseases exactly what gene causes them. We know how to create a working copy of that gene. And if we could only swap out the patient's defective gene in our cells with this working gene, we would be able to fix them. We know that we would cure them. And there are other applications for gene therapy as well. Recently, there's been a lot of excitement around being able to engineer a patient's T cells to attack their own cancer. So many theoretical things that people have been very excited about since about the 90s, but especially early on, implementation has been really, really hard to do this. So some of the challenges, one of them is delivering the therapeutic DNA partly into the correct cells safely. The most common way to deliver DNA into a patient's cells is by using a virus that's been engineered to not be pathogenic. But the virus, which can deliver the DNA into the cell's nucleus, can still cause, say, an immune response, for example, in the patient. Another challenge is integrating that therapeutic DNA actually into the cell's genome so it persists. A lot of gene therapy will just put the therapeutic DNA into the nucleus, but it won't ever actually get inside the genome. And if it just sits there in the nucleus, it will actually be expressed. You'll get the protein product, but it will only last for days, months, years, depending on the cell type. Sometimes years, more commonly days to months. And more problematically, if the cell divides, that exosomal DNA, that piece of DNA will just get lost. It won't get copied into any of the daughter cells. So their cure cannot be permanent if you don't put it into the genome. There have been ways to put DNA into a cell's genome, like viruses will insert DNA into a cell's genome, but they do so randomly in a random position. And that's bad because you can't, first of all, you can't make precise edits, you can't target a specific gene. But second of all, there's also a risk of catastrophic edits. You could put this gene into, your new gene into a place where it disrupts something extremely important and causes more problems than it solves. So, main question is, how do we target therapeutic DNA to a specific correct location in the genome? A bunch of landmark studies in the 1990s found that when a DNA molecule is broken, the cellular repair mechanisms allow you to insert new DNA at the specific site of the breakage. So this was very exciting, and the way this works, just to go into the details a little bit, if you break the DNA, both strands of the DNA, the cell has two innate processes that it might use to try and repair those. One of them is called homologous recombination. And normally in cells, this is, so the cell tries to use a functional copy of DNA to copy over to the broken double-stranded region and use that to repair it. And generally that's the homologous chromosome. So humans have two chromosomes, so if you break one, you can use the second copy as a template to figure out how to fix the first. But also, if you are a scientist trying to gene engineer something, you can provide a donor template of DNA. And if that donor template has matching sequences at the site of the break, and then a new sequence inside, then when the cell uses this template to repair it, it will actually insert that gene, that new sequence right there. The other way that cells will try and repair this double-stranded break, especially if they don't have a homologous sequence to copy from, is just by taking the two broken strands and sticking them back together. And this is called non-homologous end joining. And it's pretty error prone, so you might get deletions at that site or you might get insertions at that site, which means that any gene that's at that site will be disrupted, it could be disrupted. So one thing that I was curious about, so this is the way that we do targeted gene editing right now. One thing that I was curious about was when I was reading about this, I had heard of the concept of using homologous recombination to target a gene to a specific location before. This is a technique that scientists have been using in the lab for at least decades. And so I was kind of confused about like what is new and exciting about this. And what's new and exciting about this is that we understand now that if you use a double-stranded break, you can get a very high rate of homologous recombination. Without this, people who've tried to put like a homologous sequence and get it incorporated into the cell, generally only about 1 in 10,000 to 1 in 10 million cells actually take up that gene, which people do still use this in the lab, like for research, but it's not very useful therapeutically. So that was the main breakthrough in the 90s and early 2000s. So this is what gene editing is right now. We're trying to basically modify the genome in a specific targeted site, and the current main methodology is to create a double-stranded break at that site and then provide a donor sequence to incorporate right there. So the main questions that come out of that is, how do we even make a targeted break, double-stranded break right at the place we want to? How do we ensure that the desired donor sequence is incorporated during repair? How do we prevent other undesirable mutations from this entire process? And if we have a system that does all of that, how do we actually get it into the correct cells? So what we have found, what we have like fixed so far, we have a solution for that first question. There are 3 different technologies we use. I'm going to talk a little bit about each of them. And as for those last 3 questions, we're still kind of working on them. So this is where we are so far, basically. So the technologies for answering that first question, how do we make a targeted break? The first one that was discovered is, or developed really, engineered, are called zinc finger nucleases. They were developed in the late 90s to early 2000s. And what they are is an engineered fusion protein, which basically means that they took parts of different existing proteins and fused them together to make a new construct that was useful. So the first part of this construct is what are called zinc finger structures, which were discovered in DNA binding proteins, and there's a variety of them, and each of them binds to, in general, a specific 3 base pair sequence on the DNA. And the second part of this is a FOC1 nuclease. Nuclease is a protein that cuts DNA. That's its job, is to cut DNA. And so to engineer this fusion protein, basically you take these zinc fingers and put them in tandem so that they will bind to a specific site. And each of them bind to a specific site that's like to the left or upstream of the place that you want to cut. And this will be fused to a part of the FOC1 cleavage site. And then you have another version of that which will bind to the downstream to the right sequence of the place you want to make the cut. And it's also bound to part of the FOC1 nuclease. And the FOC1 nuclease, it requires being a dimer. It requires both halves being there to make a cut. So when, if you have both of these, when they both bind to 2 parts of the DNA, that cut will be made right there. So this was very exciting technology when it came out, again, late 90s, early 2000s. People had never had a basically modular and programmable way to engineer a cut in a specific place. If you just make a new fusion protein with different zinc finger nucleases, you can target, or different zinc fingers, sorry, in your nuclease, you can target a different region. So that was very exciting. But there were still some limitations. Mainly, it was very, very labor intensive to engineer. Part of the problem was that there aren't zinc fingers for every 3 base pair combination. Mostly they bind to GC type sequences. And there was limited success trying to engineer new ones that would fill in the gaps. And then it's also challenging to target both halves of the dimer. I read a lot of papers that were talking about what is the ideal spacing between these 2 halves of this protein. If you put them too close together, it won't cut. If you put them too far away, it won't cut. And this seemed to vary based on how big the sequence was that you were trying to bind. So it was a challenge to engineer. Each time it was a challenge to engineer. And another thing that people talked about a lot was offsite cutting. Specifically, so if you have a left half sequence and a right half sequence, ideally up there, you're going to cut right where those left and right halves meet. But if there are other parts of your genome where both left halves combine or both right halves combine, you can get cuts there as well, which is undesirable. But despite these limitations, it was still a very highly used technique and is still a very highly used technique. So in 2009, another way of finding DNA was found. Tau effector DNA binding proteins that were discovered in bacteria. And there are 4 tau effectors, and each of them, conveniently enough, binds to just one nucleotide, A, T, C, or G. So based on this, basically researchers engineered something very similar to zinc finger proteins, but with, or zinc finger nucleases, sorry, but with tau effectors in the place of the zinc fingers. But the same principle, if it dimerizes, it'll make a cut right there. And this was somewhat easier to engineer because, again, there's a one-to-one correspondence of tau effector to nucleotide. But it had its own other challenges. The DNA sequence that codes for it is very, very repetitive, and it turns out that very repetitive DNA sequences are unstable during transcription and translation and can get, like, the sequence can get reordered. So it's still pretty challenging to make new talons, sorry. CRISPR systems were discovered, or rather their capability to make double-stranded breaks was discovered serendipitously in 2012. They're used as bacterial defenses against viral attacks. And they're basically, there's a single cleavage protein, generally Cas9, that's paired with a guide RNA sequence that's complementary to where you want to make the cut. And again, Cas9 will make a double-stranded break at that specific location specified by the target RNA. And the main advantage of this is you don't have to do protein engineering anymore. Engineering a new fusion protein every time you want to cut at a new specific place was labor-intensive, it was hard, it often didn't work. But if you just have one nuclease, Cas9, and you can just swap out the guide RNA and that will target a new place, that's really, really convenient. Also, the single Cas9 protein was easier to target than the dimer. You don't have those problems of having 2 left sequences or 2 right sequences in a different place. So theoretically, this should give you fewer off-target effects. In practice, Cas9 was slightly more promiscuous than was hoped at first, but we've made improvements. But the main thing is much simpler and much more cost-effective to develop and manufacture just new RNA strands rather than having to re-engineer a new fusion protein. So CRISPR systems kind of led to the widespread adoption of gene editing in a way that zinc finger nucleases and talons really just didn't. And that's because they were just so much easier to use. So if you have any of these systems, how do you actually use them to edit cells? So this is this diagram I found for zinc finger nucleases, but it's the same principle for any of the 3. Basically, you take your nuclease, your protein that's going to make the double-stranded cut, and you actually take the gene that codes for it, and you use some sort of vector to get it inside the cell, generally a virus. And you don't need that gene to get incorporated into the cell genome, you just need it to get expressed. So you take that gene, you put it into the cell, and then the cell will make the protein from it. So it'll make that zinc finger nuclease or talon or CRISPR-Cas9 system. And then that protein will bind to the location of the DNA that it's targeted towards, and it will make a double-stranded break. What happens next depends on whether you already also provided template DNA or not. If you didn't, you just get non-homologous end joining. Those two, the break will the cell will try to repair the break just by sticking the ends together, and there's some chance that you'll get insertions or deletions there that will disrupt the gene at that region. If you did provide template DNA, you can get maybe integration of that template DNA into that region. So the limitation, again from this, it said 1 to 20% of clones have the desired mutation, which is much lower than you'd hope. And papers I read more recently sort of got that number a bit higher, but still ultimately only a fraction of the cells are successfully edited. And for therapeutic applications, that's not great. So the questions are, how do we maximize editing efficiency, and how do we select against other undesirable mutations that we didn't want from that breakage and repair process. So one thing that's commonly done for therapies, you can come in. Oh, okay. One thing that's commonly done for using gene editing in therapies is that you edit the cells, what's called ex vivo, which means you take the cells out of the human body and you edit them, and then you transfuse back the ones that were successfully grafted. This only works for certain cell types. So, what clinical trials do we have right now? Clinical trials for using gene editing.
Sarah.
Clinical trials including gene editing, so I'm going to go through a few of them. One of them is to disrupt, so one set of things that people do is disrupt an undesirable gene. For a while it's been known that there's a gene, CCR5, that people who have defective copies of this gene actually are resistant to HIV. It's because this gene codes for a receptor on T cells that HIV virus uses to enter those T cells. And there's a cool motivating anecdote of how people cool motivating anecdote for this therapy. In 2008, there was a highly publicized case that you guys may have read of, where there was a patient who had both leukemia and HIV, and they underwent a very common treatment for leukemia, which is to have all of their bone marrow cells replaced by bone marrow cells from a healthy donor. And the doctor that was overseeing this process, when picking a bone marrow donor match, found one that was both a bone marrow match but also had the CCR5 mutation just naturally. And so the doctor selected that particular patient, that particular donor, hoping that it would treat this patient's HIV, maybe. And it actually did. It worked much, much better than anyone had expected. The patient was basically cured of HIV. There was a lot of articles about them being the first person to ever be cured of HIV. And this was 10 years ago, so this patient is actually still free of HIV. They have undetectable viral load. So that was really cool, and people were very excited, but they didn't really know what to do with that information, because for an HIV patient, in general, it's just very unlikely to find someone who's both a bone marrow match for you and also has this pretty rare mutation. But now, with gene editing, one thing you could do is actually take an HIV patient's T cells out, disrupt their CCR5 gene by making a double-stranded break there and getting that non-homologous end joining pathway. So not even providing template DNA. Are you asking a question?
Maybe you should wait till the end.
Okay. Yeah, in general, probably questions, but yeah.
But like I was thinking, it's already if the HIV had already integrated in, that won't help you, though, will it?
So it
wasn't even video.
Okay, questions at the end. But this is a treatment for HIV patients, so it's preventing its continued spread throughout the body, and T cells will replicate. So you disrupt the CCR5 in the T cells. You make the double-stranded break. They get repaired in an error-prone way. And clinical trials are actually ongoing for this. So this is an ex vivo editing of T cells. So again, you take the T cells out of the patient's body, use zinc finger nucleases to disrupt that CCR5 gene, and they pointed out here they only got a median modification rate of 25%, still quite low. But since the cells are outside the human body, they can select for the ones that actually got modified, expand them, and then transfuse them back into the patient's body. So phase 1 trial, which is just for safety, was completed in 2014. This was the first clinical trial of gene editing. And the phase 1/2, which is for safety and efficacy, started in 2015, and it's actually expected to complete next month. So we'll get some data on whether it worked next month, which is exciting. So what if we want to fix a broken gene as opposed to just disrupting one? So there's an ongoing clinical trial for Hemophilia B, which is a disease, well, again, a monogenic disease in which liver cells produce a defective clotting factor called factor 9. This is caused by a single defective gene, and the therapy that's being developed for it is a single one-time injection of the gene for zinc finger nuclease plus the donor DNA, which is functional factor 9, and it's delivered inside a virus. So phase 1 clinical trial for this began in 2015. And there's sort of a major problem to overcome here, which is the editing efficiency is really low, which is like a 20% success rate. And that's, if only 20% of your liver cells are modified, that's not going to give you enough functional factor 9 for healthy clotting. So how do we deal with this? How do we get enough working factor 9 from only a fraction of liver cells? We can't do ex vivo screening for liver cells. You can't take out liver cells and put them back in. They won't tolerate that process. It will damage your liver. But what you can do is increase expression levels, and basically this means get the successfully edited cells to make lots and lots and lots of factor 9. So they did this using something called safe harbor sites in the genome, which are basically sites with high stable expression in genetic terms and always on or constitutively active promoter. Most genes are not turned on, or many genes are not turned on until they're needed, but some genes that are very frequently needed or very, very common, basically their promoter is always on, and so they're always transcribed. And the other reason this is called a safe harbor site is that it's been very well studied, and we know that the expression of nearby genes will be unaffected by adding new genes in this region. And this is actually a risk of gene therapy in general, or gene editing in general, that you might insert a gene into a place where it ends up modifying, it ends up affecting the expression levels of the genes around it and causing unintended side effects. So rather than editing the defective factor 9 gene directly, you can compensate for it by adding a new working copy in a safe harbor site. And this is why I put quotes around fixing here. We're not actually directly fixing the gene, but we are compensating for it. More clinical trials. So this is the first, will be the first clinical trial for CRISPR in the US and in Europe. It's an upcoming trial, it hasn't started yet, for beta thalassemia and sickle cell anemia, both of which are diseases of oxygen transport. The standard of care today for these diseases is to get regular blood transfusions, sometimes as frequently as once every 2 weeks, which has significant side effects. And it could be fixed by turning on the gene for fetal hemoglobin, which is a type of hemoglobin that actually carries oxygen much more effectively than adult hemoglobin, but is generally turned off after infancy. So what this therapy does is it actually uses CRISPR to delete an entire genetic region using multiple guide RNAs, shown here. And that region is responsible for turning off the fetal hemoglobin gene. So if you delete it, the fetal hemoglobin gene will turn back on and get expressed. So clinical trials are, again, phase 1, 2 clinical trials are beginning this year in the US and in Europe. So, and that's the first CRISPR trial. And in the US and in Europe, they started in China actually in 2015, CRISPR trials did. So summary of clinical trials right now. So far, there are 13 ongoing clinical trials that use gene editing in the US and in Europe. So far, all of the ones have used zinc finger nucleases and talons. CRISPR ones are scheduled to start this year. And all of them are phase 1, 2, or earlier. There are 3 phases that you need to go through before you can get the beginning of FDA approval and go into market. So yes, CRISPR trials will begin this year. They began in 2015 in China. And the primary applications so far have been monogenic diseases, things you can fix just by correcting one gene. I think polygenic diseases will eventually come, but right now they're mostly a data and modeling problem. For polygenic diseases, we don't really know where to begin about what genes we should be correcting and how. Cancer immunotherapy, engineering T cells to attack cancer is there's like 3 different multiple clinical trials for that right now. Another thing, actually, enabling this universal version of this engineered T cell therapy. Normally, the way that you engineer T cells to attack cancer is you take the patient's own T cells and modify them. But this process of modification is actually quite long and quite costly, and also you're taking T cells from a already pretty sick and vulnerable patient. So what would be great is if we could take someone else's T cells, engineer them to attack a specific cancer type, and then provide those T cells to everyone who has that cancer type. And the main reason we couldn't do that in the past is that you get graft versus host disease, where the T cells are foreign and so they attack everything in the other patient. but there's a CRISPR therapy in clinical trials right now to disable that part of T cells. And one thing that I think is really cool is there's a clinical trial starting for disrupting incorporated viruses. So for example, HPV gets incorporated into, say, cervical cells, and then it can last in those cells' genomes for years and cause problems down the line, like cervical cancer. And so there's trials beginning for HPV in China to actually delete and or disrupt the HPV that might be incorporated inside a cervical cell. Therapeutic challenges, people have talked a lot about off-target effects. There's been a lot of effort in making them better. Cas9 has been repeatedly engineered to improve selectivity, and new, more selective nucleases that can work with the CRISPR system have been found. There's still the problem of low editing efficiency. It still seems to mostly be between 20 and 40%. One company recently said that they got 80% in preclinical data, but they haven't published their data yet, so it would be cool if we've gotten there. Safe in vivo delivery has been a challenge common to all gene therapies. How do you get the DNA in without triggering an immune response in patients? And ensuring the gene editing protein doesn't persist in the cell. So if that gene that codes for zinc finger nuclease or CRISPR ends up staying in the cell for a long time and continuing to produce new copies of CRISPR or zinc finger nucleases, it basically increases the chance that down the line eventually you'll get some sort of off-target cut. And new challenges are regularly being found. This rather alarmist headline is from January of this year, where people found that some percentage, like half of people currently had immunity to CRISPR, meaning that their T cells are likely to attack cells that have Cas9 inside them, because they think those cells are being infected by bacteria, because Cas9 is a bacterial protein. When this study came out, there were a bunch of alarmist headlines like this, but when I read the paper and also read any article that actually quoted the scientists involved, most of them were like, yep, this was a pretty anticipated setback. We expected we would have to deal with this. There are a bunch of ideas we have for how to deal with this. And looking at the news cycle in the past 4 years and reading a bunch of articles about CRISPR, there's been definitely this theme I saw of some paper about a problem in CRISPR or gene editing in general gets published, and then there's just a bunch of articles about how this means it'll never work. So I think there'll be setbacks and we'll figure them out. So I will end on this less alarmist headline, which was also published at the same time as the last alarmist headline, which I resonate with. We obviously still have to wait for the clinical trial data to come out, and there will probably be more setbacks, but it still seems pretty promising. Questions?
Yeah, we can do it about it. For sure. Yeah.
So you mentioned towards the end, polygenic gene editing is something that's possibly coming. So I seem to remember reading somewhere that trying to do multiple changes simultaneously is really difficult, at least at the moment.
Yeah.
I guess, why is that? And do you think that's something that can be easily solved?
So you mean editing 2 different genes at the same time?
Right.
Well, I guess there's the multiplicative problem of you have only some likelihood of getting the first mutation, and then you're also trying to get the second mutation.
Right.
Most of what I had read was that the reason we have trouble with polygenic diseases is just that we just generally don't have a good enough model of what things need to happen to get the disease to go away. But yeah, I don't have any particular insight into why 2 cuts are harder than 1 beyond just the general selecting for 2 independent outcomes at the same time.
If about 20%, you're going to end up with what, 4%?
Exactly. Yeah, is that?
Do you know how far along studies are in China? And if a gene editing therapy is sort of approved in China, would that speed up FDA approval or not?
So the first question, yeah, no, that's fine. As to the first question, I tried pretty hard to find out. didn't, clinicaltrials.gov actually has a registry of all clinical trials worldwide to the best of their knowledge. So they called the Chinese trials also phase 1/2, which means they're being evaluated preliminarily for safety and efficacy. There seem to be promising results so far, though obviously that's only what people are reporting on. You don't actually get the raw data until after the clinical trial is over. But the main thing they're working on is cancer immunotherapy, which they seem to be making progress on since 2015. I don't get the sense from the limited amount I know from the FDA that approval in one country tends to help, especially approval from like not the EU. But yeah.
Yeah, I did. You mentioned that they would do T-cell modifications,
Yeah.
but like I realized that if you modified a T-cell that already had an HIV infection, you like couldn't really correct the fact that it already had an integrated viral genome.
That's
Because
true,
it
but
might have only limited efficacy.
so what they're doing is preventing further T cells from being entered, which means that the virus will basically spread through any new T cell that's made, and your body is continuously producing more T cells. So the idea is as your body continues to produce more T cells, all the new ones are immune, and the old ones start dying off, you eventually end up with a body that has only immune T cells.
Tremendous amount of work though. And they aren't targeting the bone marrow? Like they aren't hitting the source.
They're hematopoietic stem cells, yeah.
Oh, okay. If they're getting hematopoietic stem cells, those might not be producing the proteins that cause HIV to target it because it can't anchor, so that might be totally fine. Cool. That just confused me. I thought
Okay,
it was
yeah,
like
sorry.
a blood swap, but like if it's
Yeah.
if it's bone marrow stem cells, that's what
It
it
is,
works.
yeah. With the precursors to T cells. Any other questions? Aaron?
Yeah, you mentioned in the hemophilia cure that for claudic factor 9 and whatnot, the injection actually had only about a 20% throughput rate. Is it 20% after exposure to all cells? Can you just reapply it? Like after 3 successive applications, it sounds like you'd have a 50% throughput. Is that traumatic or expensive?
That's a really good question. I don't really know the answer to that question.
Sure.
So it's in phase 1/2, and one of the things that phase 1 trials are for are to do things like optimize dosage. So it could well be that this is like a long 5-year study. So it could well be that they might be going to try, they might be trying doing the first treatment and then seeing if the second treatment is warranted or if that would help.
Neat.
Yeah.
So even if data from China is not like immediately valuable to the FDA for, or not valuable, but like whatever to the FDA for like approving some sort of therapy, do you get effects like there'll be some sort of set of trials in China and then they're successful and then like this incentivizes people in the US to like go and do big expensive trials in the US also?
Yeah, that seems possible. I noticed that when I was reading about the China studies, there were so many articles that seemed very competitive, like China is racing ahead at gene editing, and so at least maybe in the public perception, Americans want to be up on the latest tech in the same way that China is. I don't know how much that'll actually end up influencing policy, and I think we are putting quite a bit of effort into gene editing trials just now. The FDA is being pretty conservative overall, and worried about possible side effects, but I think that's also maybe the norm for the FDA. Yeah.
So who is pushing this work? Are the big drug companies investing in it, or is this mostly government-funded university research work?
The basic research for CRISPR and zinc finger nucleases and all of those came from research labs. Primarily for CRISPR, it was the Dowdell lab at UC Berkeley right here, and another lab at the Broad Institute by MIT in Massachusetts. There's been a whole question of patenting for these gene editing techniques. Zinc finger nucleases are actually patented by Sangamo Biosciences, which currently is running all of the clinical trials on zinc finger nucleases. But basically for each of those therapies and clinical trials I was talking about, they're all backed by a startup who got an IND, investigational new drug. This is a thing that you get granted by the FDA to say, basically gives you a green light to start clinical trials. And like the specific CRISPR technique for beta thalassemia, for example, is a patent pending for a company called CRISPR Therapeutics. So basically what it seems like right now is that every company that comes up with a specific engineered version of CRISPR or something for a specific disease is trying to file a patent on it and then get it through clinical trials. Does that answer your question?
Kind of. My behind the scenes is a cynical viewpoint that big drug companies like Baxter don't want to cure patients. They want to have drugs that keep patients on the drugs for the rest of their life.
We're also out of time.
Oh yeah, we might be out of time. I will say that none of the clinical trials I've found so far actually were from big companies. They were from startups that were doing gene editing. Okay, cool, thank you.